Okay, let's finish out chapter 35 by talking about how our stems are growing. So all of the shoots, the leaves and stems, will derive from the shoot apical meristems. And that is this dome of dividing cells, actively dividing cells that is at the top of the shoot. They will be protected by the leaves of the apical bud. The shoot lengthens mainly just due because the internode cells are going to get longer. And just like in the roots, the apical meristem gives rise to three primary meristems: the protoderm, the ground meristem, and the procambium. The primary meristems give rise to primary stem tissues, and leaves develop from leaf primordia. Branching of stems is still considered primary growth. Branches and thorns come from division of the axillary bud meristems. And apical dominance is the inhibition of axillary buds as they get closer to apical buds. Let me say that again. Apical dominance is that axillary buds become inhibited as they get closer to apical buds. So removing apical buds during their pruning can remove this from happening, and a lack of sunlight and other factors can adjust the growth by increasing or decreasing apical dominance. The stem is also covered by an epidermis, and it is 1 cell thick. It also has a waxy cuticle to prevent water loss. There may also be guard cells and trichomes on the stem's dermal tissue. The ground tissue will be mainly parenchyma cells, and the collenchyma and sclerenchyma cells will be strengthening and supporting the stem. The vascular tissues are running the length of the stem in vascular bundles, and lateral shoots develop from axillary buds. The vascular tissue of the stem will converge and merge with the root. In most eudicots, vascular tissue consists of vascular bundles arranged in a ring, and in most monocot stems, the vascular bundles are scattered throughout the ground tissue.
Stem cross-sections. The outermost layer of a eudicot stem is the epidermis. In most eudicot stems, the vascular tissue forms separate strands called vascular bundles. Each vascular bundle consists of xylem toward the inside of the stem and phloem toward the outside. In most eudicot species, the vascular bundles are arranged in a ring. Ground tissue between the epidermis and the ring of vascular bundles is called the cortex. Ground tissue in the center of the stem is called pith. As in eudicot stems, the outermost layer of a monocot stem is the epidermis. However, the arrangement of vascular bundles in most monocots differs from most eudicots. Instead of forming a ring, the vascular bundles are scattered throughout the stem. Each vascular bundle, consisting of xylem and phloem, is surrounded by ground tissue.
Leaf cells will develop from leaf primordia, and leaf primordia are along the sides of the shoot apical meristem. Secondary growth in leaves is minor or non-existent. They don't need to get thicker. And the three primary meristems give rise to mature tissues of the leaf. They have dermal tissues, ground tissue, and vascular tissue. The leaf epidermis is covered by a waxy cuticle, except at the stomata, which are not covered by any wax. That way they can open up to exchange carbon dioxide and oxygen. Stomata will be flanked by guard cells. These are cells that will open and close the stomata to regulate how much water is lost through that opening. The ground tissue in a leaf is called mesophyll, and it's sandwiched between the upper and lower epidermis. This is where photosynthesis is mainly happening. The mesophyll of eudicots has two layers: the palisade and the spongy mesophyll. The palisade mesophyll is the upper layer. This is rich in chloroplasts to capture as much light as possible. And the lower layer is a spongy mesophyll that's loosely packed, so it has plenty of airspace for gas exchange. Vascular tissue of each leaf is continuous with the vascular tissue of the stem. So the veins are the leaf's vascular bundles, and they provide structural support for the leaf. The veins in the leaf will divide and branch through the mesophyll. And leaf veins will be enclosed by a protective bundle sheath that regulates movement of substances between the vascular tissue and the mesophyll.
Leaf anatomy. Let's study a typical leaf. The leaf is protected by the upper epidermis and lower epidermis, which secrete a waxy coating called the cuticle. The epidermis is interrupted by tiny pores called stomata, singular stoma, which are flanked by guard cells. The ground tissue system of a leaf, called mesophyll, is sandwiched between the upper and lower epidermis. There are two types of mesophyll, palisade mesophyll and spongy mesophyll. The leaf's vascular tissue system is made up of a branching network of veins. Each vein is composed of xylem and phloem tissues.
Secondary growth will increase the diameter or the width of stems and roots in woody plants. After the vascular cambium has formed, it can produce secondary xylem inside and secondary phloem outside. Some stem cells of the vascular cambium will produce vascular rays. And as the vascular cambium diameter increases, secondary phloem and tissues on the outside rupture because they don't grow as fast. And the epidermis also ruptures. The second lateral meristem, the cork cambium, forms from the parenchyma cells of the cortex. The cork cambium's cork cells will replace the ruptured epidermis. By the time the plant is 2 years old, the vascular cambium will produce more secondary xylem than phloem, so most thickening will be in the secondary xylem, and the cork cambium will produce more cork. Each cork cambium and the tissues it produces will form a layer of periderm, and bark is what is all the tissue exterior to the vascular cambium.
Some plants get so tall that they would topple over if they did not also get wider. Secondary growth enables these plants to increase their girth significantly to support their added height and weight. Widening occurs at lateral meristems, also called secondary meristems or cambia. We'll use a longitudinal section and a cross-section of a stem to illustrate the process of secondary growth. In this view, you can see a ring of vascular bundles between the pith and the cortex. The epidermis is the outermost layer of the stem. When you look at a detail of an individual vascular bundle, you can see that there is a layer of cells between the primary xylem and the primary phloem. This layer consists of undifferentiated cells. It is a lateral meristem known as the vascular cambium. During the growing season, the cells of the vascular cambium divide, forming rows of cells on the inside that mature into secondary xylem. The cambium cells also divide to form rows of cells on the outside, where they mature into secondary phloem. These secondary vascular tissues add both conductive capacity and support to the growing stem. The vascular cambium produces secondary xylem and secondary phloem during each growing season. As the vascular cambium cells divide, the outermost stem tissues must expand to compensate for the increased internal girth and to retain continuity of the stem circumference. Another lateral meristem known as the cork cambium develops in the outer layers of the cortex. The cork cambium divides in a manner similar to the vascular cambium and produces layers of protective cork cells on the outside. This plant has now completed 2 years of growth. As the vascular cambium produces new secondary xylem and secondary phloem cells, the circumference of the stem increases. The epidermis, cortex, and primary phloem are pushed outward, crushed, and eventually sloughed from the stem. The new cork cambium produces cork cells, while the vascular cambium continues to generate new secondary xylem and phloem. Some plants get so tall that they would topple over if they did not also get wider. Secondary growth enables these plants to increase their girth significantly.
Okay. Dendochronology, or tree age. Tree rings are visible where late and early wood meet and can be used to estimate a tree's age. And so analyzing a tree's ring growth patterns can be used to study past climate change, as thick rings will indicate a year that was warm or wet, had really nice growing conditions, and thin rings would indicate a cold or dry year. If you look at a tree trunk, there's different layers of the wood. The heartwood is the older layers. They are secondary xylem that no longer transports xylem sap. That's just called hardwood. Then sapwood is the outer younger secondary xylem that is still transporting xylem sap. The cork cambium gives rise to cork cells that accumulate at the exterior of the cork cambium, and the tissues it produces will compose a layer of periderm that replaces the epidermis. The periderm is impermeable to water and gases, although there are lenticels in the periderm that allow for gas exchange between living cells in the stem or the root and the outside air. Okay, so here's a nice little flow chart showing how primary and secondary growth is happening. As always, I'm available through email or come by my office hours. I'm happy to talk with you. Happy studying, and I'll see you later for our next chapter. Bye.